Good evening, everyone. Group 1, are you ready to present?
Yes, please, ready. Am I audible?
Yes, Siddesh, you are audible.
Okay. Muskan, just give me a minute. I'll share the slides.
Okay, sure.
Are the slides visible?
Yes, it's visible.
Okay. Mm. Can I begin?
Yes, you may start.
All right. So, good evening, everyone. Today, I'm going to present a part of the project which we did individually, but we would like to present individual parts today in the group. So the project was on human gene and to analyze it using bioinformatics tools, which we learned in this internship program. And there were total of 6 members who were in this group. So first step was to select the human gene. And among the scientific reasons, one of the reason to choose this particular gene, which we chose, was because it is of human clinical significance, but also the gene itself, TP53, was shown to be involved in around 60% of all the cancers of humans. So this particular gene was chosen. Um, so the gene name is TP53. Full gene name was tumor protein P53 with respective gene IDs, and the organism was Homo sapiens, that is from humans. And we properly documented all the related things which we found on NCBI library. And what was important is that we saw that TP53 encodes a tumor suppressor protein containing transcriptional activation, DNA binding, and oligomerization domains. So it goes and binds to these particular domains which are present in the DNA. The role is in cellular stress to regulate the expression of target genes, which in turn induces the cell cycle arrest, apoptosis, that is programmed cell death, senescence, DNA repair, or also some changes in the metabolism. And if there are any mutations in this particular gene, it leads to variety of human cancers, including hereditary cancers such as Lee-Fraumeni syndrome. So this particular syndrome is very famous and has been well documented and studied. The second step was to retrieve the gene sequence. And for this, I used again the NCBI nucleotide database. The FASTA file was downloaded, as you can see on the right hand side. Um, and it had the respective accession numbers. This is just for the reference. And the sequence length was around 20,000 base pairs. This is the representative of what I saw and used on the NCBI website. And therefore, the TP53 was found to be appropriate, which means that it actually had the gene sequence, which was not partial, because some of the genes, if you go and choose them on NCBI, it tends to have partial sequences, then it is not quite useful. So since we found full gene sequence and we knew the functions which were related to it, this sequence was chosen and was used for further analysis. And now here on, my other colleagues who have done the respective steps in the project will present. I will continue to control these slides.
Yeah, thank you. So after selecting the TP53 gene, the next step was to understand its sequence and record the available gene data database. A sequence record is like a identity card for the gene. So it was very important to know the biological information and the sequence. So the first thing we observe is the accession number. Um, can we go to the next slide? Yeah. So the first thing we observe is the accession number, which in our case was U94788. This is a unique identifier, so researchers all around the world can retrieve exactly the same sequence. And the next we saw the organism, which was Homo sapiens, and we confirm it belongs to humans. The molecule type we used was DNA. It tells us that the record contains the genomic DNA, and it was not any RNA or protein. This also contained a feature section where the biological where we can see biological annotations or identify the location of the TP53 gene, the coding sequence or the CDS, and all other important sequence features. Uh, Identifying the sequence record is important because it helps us to verify uh, that we are using the correct gene from the performing any process or any analysis. Uh, can we go to the next slide? Yeah. So once we confirmed that the correct TP53 sequence, the next step was to perform the blast analysis. So as we all know, the BLAST is a basic local alignment search tool. It compared our query sequences with millions of sequences stored in NCBI database to identify similar sequences. So since our query was a DNA, we used a BLAST N, and which compared nucleotide sequence with a nucleotide database. Um, can we go to the next slide? Oh, let it be. Like I need the picture one. Yeah, the best hit was the human TP53 gene itself. It showed 100% query coverage, 100% identity, and an e-value of zero point 0.0. So it confirms that the retrieved sequence was correct. The first this in this column we can see the organism. This tells us which species uh, is the matching sequence, like matches the most. The next one is query coverage. Query coverage indicates how much of our sequence match the database sequence. It is 100%, means the entire sequence was aligned. Then we had a percent identity. So it shows how similar the sequence are. The higher values indicate the higher similarity. And then we had e-value. So e-value represents the probability that the match occurred by chance. So the lower values are better. So next point, next slide. The best match was like from the class we got to know it was mus musculus, which is mouse, and its e-value was zero point 0. So overall, the BLAST analysis confirmed that we selected the correct TP53 gene sequence and demonstrated that this gene is highly conserved. Like it has very less mutation in evolution. So, uh, Yeah, that's it from me.
So, just before we continue, I would like to state here that since everyone has done their own steps and this was not necessarily in coordination with each other, like everyone submitted their own file, the best match can vary a bit. So, don't wonder if this happens in the next slides. All right.
Hello, am I audible?
Yes, Vaishnavi, you are audible.
Okay. Uh, this is not the slide. I
The
think next slide
one more
is
next
mine.
slide.
Okay, later we perform the retrieve of homologous sequences, in which we have picked out the five homologous sequences which have the common ancestry having the same genes which have been conserved or which are the same sequences in these five particular organisms. So we picked the five organisms which are showing highest score. Next slide. And this is done result of the blast. Next slide. And later we performed pairwise sequence alignment by in which we have performed one with the human sequence and other with the best matching non-human sequence, which is a chimpanzee. And for this, we have pasted our first. We have performed this process and later. Next slide. So this is the result of that particular. So as we have seen the length and the identity similarities, which is 91.1% match, and the gaps which are showing the insertions and the alignment score, which is 14813.5. And according to this data, the non-human match and human is highly similar because it both of them T53 gene it's showing highly similarities. Uh, it is showing the common ancestry and which is essential tumor suppressant gene. Uh, this is the blast findings which we have got. Uh, Next slide. Sorry, this is the blast finding which we have got from comparing two different alignment by using Needleman Wunsch. Later, the multiple sequence alignment in which we have performed by taking the first five organisms which we have earlier selected, and we have performed cluster omega, and the results were there. There are some conserved region all across the uh, sequences. There are some portions which are consecutively similar. So they showed the TP53 gene has been conserved during the evolution, and these regions are naturally get selected during the evolution, and during the period of time they they were similar in all these five organisms which we have been selected as top hits, which have the similar score, which have the high score with comparison with the humans. This is it from me. Next.
Hello, hello. Is my sound clear?
Yes, Ragini.
Yes. This, this is slide 8, guide tree analysis. Using guide tree and phylogenetic trees to reveal evolutionary relationship among a species. Next slide. Group first, human and chimpanzee clustered together as the most similar pair. And the next most similar group, human chimpanzee closest sequence matched in alignment. Next, most distant cow sequence most divergent from primate group, and the observation is matches blast results. We go to next slide. This is phylogenetic tree, which is closest relatives. Human and chimpanzee share the most recent common ancestor. Most distant is cow is most evolutionary distant in the analysis.
Ragini, you are not audible.
Good evening. Am I audible, ma'am?
Yes, you are audible, Saram.
Okay. After analyzing the TP53 gene sequence, the next step was to study its protein product using the UniProt database. The protein encoded by TP53 was found out to be cellular tumor antigen P53, which is one of the most important tumor suppressor proteins in humans. Uh, by using the UniProt, we also found out that the protein consists of 393 amino acids and has an annotation score of 5 out of 5, which indicates that the information available for this protein is highly reliable. Uh, the evidence level is listed as evidence at protein level, uh, which indicates that the existence of this protein has been experimentally confirmed. Now, functionally, P53 acts as a transcription factor. It regulates the expression of several genes involved in maintaining genomic stability. When DNA damage or cellular stress occurs, P53 can induce cell cycle arrest, giving the cell time to repair the damage. If the damage is too severe, P53 can trigger apoptosis or program cell death, preventing the spread of damaged cells. Uh, Next slide, please. So after confirming the protein characteristics by using UniProt, we verified the nucleotide sequence information uh, across major international nucleotide databases so that we can ensure the consistency and authenticity of the data. Uh, for sequence verification, the TP53 sequence was searched in three major nucleotide sequence repositories, that is NCBI, DDBJ, and ENA. The accession number retrieved from our analysis was searched in each database. We observed that the same TP53 sequence record was available in all the three databases with matching sequence information and annotations. Although the databases are maintained by different organizations in different regions of the world, they synchronize their nucleotide sequence records regularly. Thus, researchers can access the same sequence data from any of these databases. So this verification step ensured that the sequence used throughout the analysis was accurate and authentic. Yes, thank you.
Thank you, Sarah. So, moving on to the last part. To summarize the study we performed, we have been given to certain questions for which I have stated few points in the slides to explain it further. So, first, the biological function was been actually studied still since the beginning. That it has a major role in protecting cells from genetic damage. It acts as a tumor suppressor by really acting regulating cell cycle progression, DNA repair, and program cell death, which is also known as apoptosis. The second point was that uh, it has closest relatives to humans. So, through the phylogenetic analysis, we found that chimpanzees had was showing closest evolutionary relationship with humans. Also, bonobo, which also showed highly related, tightly linked genetic pairing with the chimpanzees. Uh, the next one is uh, highly conserved regions were found through multiple sequence alignment. which reveal that there are many regions across the species which had very similar regions. Next slide, please. Okay. Why these regions are conserved? Since TP53 is involved in essential cellular processes, mutation is important functional regions which can be harmful. So these regions have remained largely unchanged throughout evolution due to natural selection. Then So yes, using all these databases such as BLAST, Cluster Omega, Phylogenetic trees, etc., it provided consistent data and among all the sequences. This shows that accuracy of the identified evolutionary relationships and functional interpretations. And then So taken together, the findings indicate that EP53 is both functionally important and evolutionary conserved. The high level of conservation across species highlights its fundamental role in cellular protection and genome stability. So coming to a conclusion, overall, this study demonstrates how bioinformatic tools can be used to investigate gene function and evolutionary relationships. This analysis not just provides insights into biological importance of the gene, but also shows how sequence conservation can reveal significance of a gene across different species. And with this, we end the presentation, and we are thankful to our guide, Ms. Muskan Kashyap, and our team members.
Okay, very good, Group 1. Your presentation was very nicely done. Does anyone wants to ask any questions to Group 1? Any questions from anyone? Okay, then I would like to ask some questions. Okay, so my first question is, even though Siddesh already cleared that the sequence would be different, but why there was the mouse sequence first and then the chimpanzee? Why is this difference?
So, Muskan, should I answer or?
Yes, yes.
Okay, so what happened is, if we let me take you back to the first slide. Second. So what happened is, so we took the gene sequence, but when it comes to particular search on the BLAST, if you look at the description table here and you go from the description table, you can find many organisms which are there here. So the first thing was that I first of all skipped the first point because I took the database based sequence itself. So the first sequence is going to be identical, and the other organisms are like written here, but they are not necessarily grouping together the closest organisms. So the right way to do it, in my opinion, would be to look at taxonomy here. So that is why the approach I also did, and what others also did, was to go with the taxonomy table, and here we can see which are the actually closely organisms. Although I would say that mus musculus answer was also not wrong, that it was close, but I would say it is not the closest one. So musculus is somewhere down here, so it's not listed here, but it's definitely down there because in the description table it lies down somewhere. So I cannot see it here in the snapshot. But yeah, this is the reason why in some approaches the description table it was mus musculus which was the closest, and in some approaches the pan troglodytes, which is chimpanzee, was the closest.
Okay. Okay, that's good. Okay, and one more question that I would like to ask. You mentioned that the gene that you selected, it caused a variety of human cancers, and there was one famous syndrome.
Yes.
Yes, so can you let me know a little bit more about that syndrome? Because you mentioned it was very famous.
Yeah, actually, I have. I'm not a cancer biologist, but so basically, it is it actually is caused by the mutation of the tumor suppressor gene, and and it has the lifetime risk. In 90% of the cases, it is for women, and 70% is for men, and so basically, there are different types of cancers associated. So there are sarcomas, which are related to soft tissue. There are bone cancers, which is osteocarcinoma, and there are also breast cancers and brain tumors. So it causes different varieties of cancer. So Lee-Fraumeni syndrome is hereditary, but it is rare. Yeah.
Okay. Okay, my last question is, can you open the accession number of your gene?
Like on the website or
From the picture that you have pasted here, where you were showing the
okay.
accession number.
Yeah, yeah. Just a second. Yes.
Yes, so here the accession number is starting with U, right? U94788.
Yeah.
Okay, so there is U written here. So is there any specific meaning of U in this accession number?
Mm, I
Can I
actually
answer?
am not aware at this moment.
Yes, Bhargavi, you can answer.
Yeah, so it indicates that like I guess it indicates it is a direct nucleotide sequence, like in gene bank.
Okay, very good. Okay, so these were the only few questions from my side. If any other student wants to ask any question, you can either ask or you can type in the chat box as well, because the group one gave a very nice presentation. All of the steps were very nicely done. So any student want to ask any question? Okay, so group one, I would like you to submit your project on the Google Classroom. Okay, some of you might have already submitted it, but not all of you, I think. So submit your project on the Google Classroom, okay?
Yes.
Okay,
Muskan,
then.
can we submit the assignments till Friday? So before 30th, is that fine?
Yes, before
Like,
30th. Before
okay.
30th. Yes.
Okay. Thank you, Muskan.
Okay, then. Have a good evening and very well done.
Thank you, everyone.
Thank you, everyone.